people there. Okay. Anyway, thanks for presenting the department there. It's a nice place to go next time. And today we have got a guest here from Shishumbana Botanical Gardens. He came from China, but not directly to here because he came to the phylogenetic course in Krumlov. So welcome, Sasith Tharanga. And I would say the floor is yours. Yeah, the presentation is about herbivores, predators, relationship, and interactions.
Thank you for the introduction. So, Sasith Tharanga coming from China, XTBG. I think most of you know the place. Here I say plant herbivore predator tri-trophic interactions. But actually, so far I'm just studying them separately, like plant herbivore and herbivore predator. So, not analyzing any tri-trophic things yet. So. This is the XTBG. Shishumbana is the China, Yunnan, Southwest China. And the garden is in somewhere here, it's more border of the tropics, close to Myanmar and Laos. Yeah, so it's mostly tropical vegetation. So this is my study system. Plants, herbivores, at like many caterpillars and butterflies or moths, and that they are predators. So my talk will be about these three relationships: herbivore defense against predators, like approximatism and mimicry, and herbivore damage and plant defense. This part is kind of ongoing project. So I'm a postdoc now there. So my work is, what I'm presenting here is like PhD, part of PhD and ongoing postdoc research. So it will be two parts and a little bit of what's going on. So first section, predation prevention strategies of tropical Asian butterflies. So I think we all know about approximatism. Animals use colors for displaying their danger or warning signal, which we call approximatism. And this is the mimicry relating to butterflies. So if I briefly explain, we have toxic plants and non-toxic plants. And butterflies, that caterpillars eat toxic plants, accumulate toxins, and then they are defended against predators, where birds learn to reject, right? I think all of you know this. And then there are butterflies feeding on non-toxic plants, and some of they mimic the signal. So the mimicry. And in my study system, I selected tropical Asian butterflies, and then I remove smaller ones, and among the medium to large size, I separate them like toxic and non-toxic butterflies, mostly based on previous studies. And then the toxic ones, those who have signal, they are aposematic, and they are not toxic but not non-signal, so they are non-aposematic. And among them, some are mimicked by other butterflies, so they are models, and there are some toxic but non-model species. So I use these groups for the study. And these butterflies are distributed in mainly like tropical Asia, India to Japan, I would say. Some of them actually can be found in Australia and Africa as well. So most of my field sites are in Sri Lanka and Southwest China and also South China here. So what the relationship between toxicity, predation, or the protection against predation and their warning signal? These things have come to kind of equilibrium, but they continue to be changed, I think. And also the distribution of these signals, group, species are important. So what we know about this relationship, we generally known is like owner's signal hypothesis is two-dimensional. When the conspicuousness increase, that means the toxicity increase. They have owner's signal. The other hypothesis is the trade-off hypothesis, where it consider the protection level. So, for example, when the highly toxic ones have a less conspicuous signal, but they get higher protection, and less toxic ones have highly conspicuous signal and they get equal protection. And those have low toxin and low conspicuousness, they are less defended. That is the trade-off hypothesis. So here I introduce another dimension, the predation, partially the predation pressure change. So predation is the main selection here, right? And what happened when predation pressure change? So I had this, and I measured the predation pressure on the toxic ones, epistemic ones, mimics, and non-mimic ones. And then I measured the toxicity extracted from insects, butterflies, and used in mice, which we call LD50. And then I quantify the warning signals as using this model as the bird see it. So this all shows like this is what we see as human, right? And it's quite to comply with what birds see. And we have a gradient of conspicuousness. This is something different from, I think, neotropical butterflies. They are mostly all conspicuous, more or less. But here, they are highly conspicuous to like very close to camouflage. We can separate three groups or three mimicry rings actually. But their toxicity, they are all toxic enough. There's no significant difference in toxicity. So what did I find? We found a very interesting strong predation gradient. Actually, when I was proposing, there were a lot of people argue like maybe you can't find such a predation gradient. And while I was analyzing at the same time, the predation caterpillar paper came out. Yeah, I was writing this. So we had evidence like there are large predation gradients. So what happens? Their benefit or the protection change along the predation gradient. The red dots are the model toxic ones, and the black line is the mimic. So one message here is mimic get higher attack even though they mimic than the original epistemic one, and their protection change along the gradients. So we propose the hypothesis that this is a mechanism, this may be a mechanism that maintain these different signals in the same place, because these butterflies stay in the same place. In different locations, they have different benefits or different protection. Yeah, this is what I just said. Second part is more or less about the behavior. It's like we found, it's based on my naturalist observations for a long time. I found like, okay, they stay in different places in the habitat. So what's going on there? So as I showed you earlier, we have these three groups or three butterfly mimicry rings. We name them based on their most common genus. These are based on previous work. So this is the toxic one. This is the underside. This is the upper side of the wing, top row. Second row is like underside of the wing. These are some of the mimics. So most of these mimics, they have the mimic signal only on the upper side. Underside is more or less camouflage. So yeah, and we believe, like naturalist butterfly watchers believe, these guys fly lower and mimic fly lower in the habitat. So we know birds come from the top. We have this stratification. And if I go to another step, here the left-hand side is the upper side of the wing, right-hand side is the underside of the wing. So you see these butterflies, toxic epistemic ones, they are left and upper and underside of the wings are more or less same. And some of the mimics, they do mimic like a near perfect mimic both sides of the wings, upper and underside. But some, as I shown you earlier, they have the warning, the mimic signal only on the upper side. And here there's one group of butterflies. They have different behavior. They have warning signal totally on the top, but they do have the mimic signal on the partly on the upper underside. They can, if they want, they can open it. For example, in these open areas, when they're feeding on flowers, they open and they show the warning signals. And in some other habitats, they close the wings and can become like camouflage. So I developed this hypothesis. Bird come from above, and toxic aposematic ones can fly everywhere. They mostly fly higher. So, and mimics fly lower. They can get benefit from their camouflageness. And then we had the question, okay, but some of the mimics, they don't have a camouflage wings. We retain both upper and undersides are mimicking the signal. So yeah, it complies with our normal observations. So I developed hypothesis. The species with underside camouflage, they stay under the shade where they can close the wings and become a cryptic camouflage when they want. And if they want to show the signal, they can open the wings. Other butterflies with both mimic signal on top and bottom, upper and lower of the wings, they mostly stay in the open areas. So, and I developed some alternative hypotheses. Because mimic need protection from models, they tend to stay together. These are like classical hypotheses. They stay together, so there's no stratification. And then we have our fourth dimension, the predation gradient. What will happen along the predation gradient? So maybe any flight height difference could be because birds fly at different heights in different areas, or maybe vegetation govern the height of butterfly flight or the bird flight, and also can be the house plants. So we tested those alternative hypotheses as well. And for houseplant heights, we collected butterfly houseplant data for like very long time, or other people have been documenting them for like 100 years or more, 150 years. So we have all this data, but when you do sampling in the field, we use like plots. We don't find all the plants within the plot. So those plants we did not find in the plots to measure the heights. I used the literature data of plant heights, and they are highly correlated. So we found evidence that all three groups mimic fly lower than their model. That is statistically significant. And another thing we found was model flight height increase along the predation gradient. These things create like a new hypothesis or new questions, and mimic fly further low, it go down. So maybe mimic is using more camouflage, benefit of the camouflageness in the high predation. It's not there to show the signal. And yeah, these two are the main findings there. This is the raw data, where these points show the butterflies we observed. Another hypothesis was how they use the shade and open areas. So here the percentage of shade is 0 means totally open to closed canopy or shade. Yeah. So toxic ones are everywhere basically. They did so statistically different. These guys stay a little bit more open, I think. Yeah. But they can stay everywhere. And so we have these three types of mimics. Those who have mimic signal on both underside and upper side of the wings, and the one partially mimic on underside, and the signal only on the upper side. This is like a closed wing. So closed wing, the butterflies with camouflage underside, they mostly found in shade areas. And other two groups could be found in open areas. And our alternative hypotheses were not supported, like no relationship with the tree height or bird flight height did not change along the predominant gradients. Yeah, these two are the conclusions. I think these give us some direction why butterflies, some of them have signal only on the upper side. So need more investigation. So another thing I wanted to test is, okay, this was like experiment in the field, in the nature. We observe predation also on the butterfly wings. So I want to do like a manipulative experiment. We design this with the dummy butterflies. And we try. I think some of you are familiar with these kind of studies. So we printed these butterflies and tested their reflectance. We use both. There we have bamboo worms instead of mealworms. In China, we have people eat bamboo worms. They're quite expensive, like a luxury food, but you can buy it. And we use bamboo worm, and also we try both methods. So these, and I use this green butterfly. This is like a experimental, like a testing the method, green butterfly for control. And we place them in the forest and open areas above 1 meter height. We try another thing like hanging butterfly on strings. Method is nice, but it's a lot of labor consuming. So there I plot the attack rate on these three groups against the control, similar to this background predation here. They show more or less similar pattern, I think. Like these two increasing predation pressure. And in most of our analysis, we found this intermediate group. They either don't show a pattern or they are just flat. I'll skip these parts. So the second part is about herbivory, the caterpillar herbivory on plants. I did this work in Guangxi forest. So here I recorded 5-6 different types of damage on leaves. These forests are like karst forest, and the trees are not very high, so we can reach the canopy with a pole cutter, like 15 meters maximum being reached. So this is the South China in Guangxi province. And that time I was collecting sampling insect also, and then the COVID came and we had no access. The experiment went a little bit, could not complete the insect part, but herbivore shows along the vertical gradient, there's a increment of the herbivore. And this is same for all three ontology, like seedlings, saplings, and trees. And also I tested whether the plant height, yes, with the plant height it increased. With the crown size also herbivory increased, but DBH did not show any relationship. These are like distribution of different guilds. Maybe it's more clear here. So chewing insects increase with the plant height, and skeletonizers, they did not show any pattern. And gold makers, neither. And sap-sucking insects had decreasing herbivore rates along with the increasing plant height. Same pattern for this is leaf rollers and leaf miners. I did not check what other people have found on these. Yeah, and this is what we found. This is like the herbivory on different leaves. Yeah. So summary is just what I explained. So these two are the like completed projects and either published or under review. And here The second, another question is my ongoing like a postdoc research. In the garden, we have exotic plants, we have native forest, and we have native plants planted in the garden. So it's like a common garden for a mix of exotic and native plants. So I wanted to see how the native insects move to exotic plants, how the herbivore change. So one hypothesis was native plants get higher damage than exotic plants. And another hypothesis was the plants from neotropics have less damage than the paleotropics because the Shishombana is in paleotropics. So it means like the distance negatively correlates. And the time of interaction is positively correlated. If the plants were introduced recently, they have less damage. If the plants were introduced a long time ago, local insects adapted to them, right? And then they start eating the exotic plants. And so then all the exotic plants should have higher herbivore damage. The second one, this is about the traits. This we are still measuring herbivore traits and the plant traits. And so this is the study area, botanical garden, and we have forest. So what we found, actually it's like part of the data, and this year also we continue this study, native and exotic. Exotic plants have less damage. Yeah, it's comply with the hypothesis. The chewers and subsuckers also show the same pattern, but it's the chewers contributing to the data. And the next, heliotropics and neotropics. Neotropics had lower damage. Is that what we hypothesized? Yeah. Okay. And the chewers and subsuckers are the same pattern. We have Africa also. Yeah, here we have it. We need more. Then we try to separate them like, I don't know, continent or Southeast Asia and Asia we separated because the garden is in East Asia, Southeast Asia, and the African plants and the American plants. So they are different, but maybe we need more sampling. And this went the other way, I think. Yeah, older the plants get, they're like a lower the damage. So do you have any explanation for this? The new plants introduced to the garden get higher herbivore damage. The older plants get less damage. But theoretically, like invasive species and these enemy-free hypotheses, these things explain. Yes.
Could this be because the plants had, as they are introduced to a new environment, they are kind of shocked, you know, their fitness gets limited by the fact that they have to adapt to a new soil and stuff like that. While as they stay there longer, they have time to develop anti-herbivore strategies. Is that possible?
Yeah.
Or you were asking, I'm not sure.
Yeah. So this idea, right? The new ones are weaker, not adapted to the environment. It's not very established or uptake from nutrients or climate and all this. But the theory is opposite. Yeah, the enemy-free hypothesis is when the new plant introduced in a plant introduced to new environment, they are focusing on, okay, native insects are not adapted to eat this. They cannot detoxify the chemicals, or now it's a different test. So, and its movement happens slowly. So the new plants should have less damage, heavy damage. This can be understood from invasive plants, right? I think this theory comes from invasive plant studies. So that's why plants become very invasive. At the beginning, they spread a lot. And with the time, native herbivores start moving to the plant and it control it. So invasiveness drops. Yes.
Could it also be because the insects simply didn't know that these plants are not appetizing or not good, you know, because but at first you place something there, they don't know it.
Yeah.
seems appetizing, they eat it. After that, they realize it's not so good. So over time, they develop avoidance towards that specific plant.
Yeah.
Have you?
I don't disagree with you. I think something like similar happened at the very beginning, right? When you introduce the plants, then maybe like first two, this you can't say it's a hypothesis, right? But yeah, once they establish, so maybe it should be like kind of like a curve, bell shape, or right? At the beginning is low, and then everyone start eating it, and then plant adapted, or yeah, not very flat curve. Yeah.
Maybe here I'm not getting quite so by established. These are the plants which were planted to the garden. It's not like they would
Naturalized.
naturalized. So this is the
Yes.
newly planted tree species into the completely new conditions,
Yeah,
not naturalized like invasive thing.
yeah,
This is several individuals. So this is like those. So they planted 20 individuals 20 years, 70 years ago. This is what the graph shows. Now that second on the left.
this one, right?
Yeah. So this means that they planted 20 individuals,
These are in species.
of 20 species.
Yeah, yeah. Mix of species
But
in this one.
maybe how many individuals were?
Roughly like 5 individuals.
Yeah.
Sampling is going on like so. Now we want to like increasing more species. Yes.
Maybe to throw in another theory, maybe
Yeah.
just some survival bias in trees that are still surviving that were planted like 60 years ago. They were
Yeah.
like
Yeah.
super fit trees,
Yeah, yeah,
and so you have like a little bit of effect there.
yeah, yeah. I think we can combine these two ideas. That was my explanation also. This is very flat view, right? You need to think about in more dimensions. So like 50, 60 years old mean like even maybe mango is there. Mango is like 100,000, we don't know when it was introduced, but so we remove actually some of those like tamarind, mangoes, like we don't know when it was introduced, but we know they are exotic. But yeah, that's the case. Garden keep replacing plants, right? They maintain the garden. So what happens, these maybe, they keep introducing, but we have the exact date they planted all the plants in the garden. Those survive 40, 50, 60 years. They are adapted to environment stronger, all these things you mentioned. Yeah. So we combine all these idea, I think it's explained.
Can I ask, how old are the trees you are testing? Like, it's the same age of for all the species, or?
No, actually, it's like
Because-
it's like this age. Oh, sorry. No, it's even not normally distributed. I
Because
think.
the the newly planted species are like 1 year or 2 year old tree, and it can be more damaged by the herbivore because it's like pressure or something.
Yeah. Actually, we did not. I did this like a student project, like a training course project, part of this, but now we are continuing with more data. So we remove like, very new plants, maybe like less than 2 years, because they were not established. Some of they die, like that. Yeah. But this includes maybe 5 to 10, something like that. Very early plants we did not include. Yeah, probably we need more sampling and more dimensions of analysis. Yeah.
I want to ask, of the exotic plants you were investigating, was it only trees or also shrubs or vines?
These were all woody shrubs and trees.
Okay.
Yeah, shrubs and trees. Yeah, we need to improve this. See.
As everyone is asking, I will already make my question. Aren't you mixing there the age of the individual and when that species was introduced in China?
Oh, yeah.
Because the anime tree
Is like what the area
about when that species, the fauna and the plant species got imported,
Yeah.
not that one individual plant. So I think you can't have expectations from the enemy-free hypothesis on
By
an
the
individual age.
Yeah. Here the date is like the first introduction date of that species to the garden. But we could not find data for when it was introduced to China or the Yunnan province. Still, we could not find a source, reliable source for that. We only have what was introduced to the garden.
Okay.
So this species were introduced there. Anyway, garden was slowly growing. Most of the plants, international collaborations, all these happened recently. Yeah. One thing is the garden is very isolated from rest of the China.
Mhm.
And they got railway like last year, I think. Yeah. And the airports came maybe, I don't know, 15 years ago or something. Yeah. So what is here? These are some of the traits. Here the hypothesis was because in this area we don't have mammals. They were hunted out. No mammal herbivores. What we have is like insect herbivores. And so I hypothesized plants that were adapted to mammal herbivory. They get higher damage here because they are less dependent against the influence. So we roughly consider this is very arguable, but latex is mostly against the insects, and spines are mostly against mammal herbivores. Yeah, hairs can be both, but the hairs we measured are probably against the insects. So in the garden, there's a difference. The plants with latex, they get less damage because they are dependent against insects. And plants with spines, yes, they supported our hypothesis. They get higher damage in this area. Is the hypothesis and clear?
Can I ask, what does it really mean with about the more damage for plants with spines? Does it mean, is it like mutually exclusive, the defenses, that like plants that hair, they didn't have spines or?
Yeah. For the analysis, plants with spine which does not have latex.
Oh.
Yeah.
So, all right, so is there like, were there plants that, the samples, were there plants that had neither latex nor hair nor spines in the analysis? Or was it just either latex or hair or spine?
One of the data set is it like this. So in the data which we say these plants have spines, they don't have latex. I mean, if the plant has both latex and spines, it was not here.
So that's why you had a significant result for increased predation of spines, because you didn't have like, because if you had
something in between.
Yeah, if you were comparing it to normal situation, the spines shouldn't act as like, an increase of predation, because it's just, it's a non-functioning defense. Which means it shouldn't increase predation.
Yeah, this is like, I got your point. We will add more plants.
Don't have to, I think the experiments fine as it is, it's just a little bit misleading to say, plants with spines had more damage because why should spines cause the damage? Why should spines lead to more damage?
You mean why spines cannot defend against insects, right?
Yes, but if they cannot defend, why is it increased, right? Why is the predation increased rather than just not significant or something? That's what I meant.
Okay. Yeah. So we need more intermediate cases, I think.
Yeah, and also you need to measure it on the continuous scale, I would say, like
Yeah.
if you latest yes or no.
Yeah.
There must be
This grouping
a
is
huge amount of variability between yes latex.
Yeah. So this was like a short time study, kind of preliminary actually. It's like a 1 week sampling, with the students.
During the XTBG.
Yeah. FX.
Yeah.
And I asked, they need mini projects. I say try this.
Mhm.
Yeah. So we need to make it like a large scale with more sampling. So some of the ongoing projects, Second part of this exotic plants, native herbivore thing is, I am thinking to test how the plant affect herbivore, the herbivore's morphology or their genetic differentiation, any expression differences. This is still like maybe this year we started. Another project, I won't say like I don't have students, I'm a postdoc there, and in Chinese system, postdocs don't get to supervise students. So most of this work just done by me. And so here, the pollinator, herbivore, and plant networks. For this study, I chose South Asia, Sri Lanka, India, Bangladesh, and Nepal. And so I am like collaborating with institutes there. And we apply some funding, still no results came out. So it's going on like a student projects again in these 4 countries. And some of the data is being analyzed. Another small project is organic farming. Now, I'm from Sri Lanka, and we had a very disastrous decision of moving to organic farming, like 100, I don't know how, maybe 70% move to organic. It was like plan was to gradually move by 2030, but they decided to do it very fast. So yeah, then we started this experiment. Yeah, that's all I have to share today. Thank you. More questions? We talked some of them. We have time. More questions?
I don't know, Leo has questions?
Yeah. In the study, we're showing about the effect of tree height on the herbivore. Did you test that for different species or for different separately for the species, or you put everything together because different species are in different heights? So how much is that an effect of the species?
Yeah.
And how much is it an effect of the same species being eaten more or less along the elevation gradient?
The result I showed you include all, and I just use the species like a random factor.
Okay.
But earlier we did another work I did not show here. It's like published work using seedlings below 50 centimeters, right? There we separate like species. We had enough sampling, so we pick the common species. Still show the same pattern actually. But yeah, I would let's separate the most common plants. That's what we want to do. Analyze. Yeah.
But in that analysis, you did
all together.
You use the species as a factor.
Yeah.
Oh.
Any suggestions? Any questions? I know you guys are expert all here, herbivore, insects. Yeah.
No, the herbivore was on the elevational gradient. The x, the 1,500 was the meter elevational.
No,
it was
the height of the
height
tree.
of the tree.
Yeah, in centimeters, sorry.
And there were no higher trees above that. All of them are, there are.
Yeah, this forest is very short. It's like very windy.
I mean, 1,000. It's like to my belly.
No, 1,000
15
centimeters.
meters.
Oh, 15 meter. Aha. Oh, okay.
It's not the emergent, but up to
Yeah.
like a canopy.
Okay, there was 15 meter, not. Yeah.
Not like real. It's subtropical.
I saw 1,500. That's why I was like, oh, this must be in centimeters. Yeah. 15 meter. Okay, that makes sense. Yeah. Good. Maybe how do you do this plant pollinator networks? Do you record them? Do you watch them, or what's your methodology?
One is, we have like students observing this in all these countries. So they rear butterflies or caterpillars, and others like regularly. I give them some number of species with many individuals. So, so they also continue to record the pollinators and some other record abuse on them. And other data is like literature reviews, and I collected for a long time, all these together. So our target is to see like which species we get more data. They also have some experiments with native plants and exotic plants. We have a few minutes. No butterfly question?
Yeah, that's one question. So in the experiment where you had a black butterfly, like you measure also like reflectances, like
Oh yeah.
because you say it's like a conspicuous butterfly, but maybe to the bird it's something they can recognize. Not a human vision color, but maybe some.
They do have these UV reflecting small dots. We see like white dots. Yeah, I measure UV reflectance and all of that. Size and shape. Yeah, so integrated into the calculations, and then we got the conspicuousness.
The mimics, I suppose they were Patesian mimics.
Yeah,
Because they were unpalatable, or they were palatable, or
mimics are unpalatable. Like they have co-mimics, the Mullerian mimics.
okay, they are.
Yeah, they have rings.
So all of them were Mullerian, so all of them were unpalatable.
Yeah,
Oh.
because I collected it's all the species of Mullerian mimics. We definitely miss some Bayesian mimics. You can't. They are very rare, most of them. But you test the toxin with the Mullerian mimics, right? And I include all of them. But I tested toxin at each species level separately, and then we plot national species.
Does the toxicity increase with display?
No,
So it's
yes, no relationship there. Not different.
Very practical question. The plasticine which you put to the butterfly, you put it like a body to the middle. With the models, and you record the bite marks on the plasticine body.
That's what we expected, but in practice we had different situations. For
What I
example,
would expect.
so we use kind of a base as a plasticine, and we put the mealworm on top of it, not mealworm, bamboo worm, stick. So in some cases, they took the worm removed or partly eaten, and yeah, there were a lot of damage to the wings also. They attack the wings.
Yeah.
Yeah, they try to grab it, and or they take it and drop somewhere. All these scenarios were there, so
Yeah, I
to
would
figure out.
expect the wing scenario. That's why I was wondering why you're putting the plasticine in there. So yeah, I don't think the birds would be aiming for that. But that's good that you recorded all of them.
Yeah. So in previous studies, like aviary studies, people put something printed or they put the food on it, right? So this is like we give the butterfly wing and the food both. Well, we use glue and we stick the bamboo worm. It stay there like 4 days live, like a little bit moving, so it's more like very live. And we did another, yeah. So there are a lot of things going on, but mostly birds. Yeah. So in analysis, I don't know, need to think more and modify, or either put one of them. The difficulty is damage to wings, because we printed underside, upside separately and stick together. So the wings are kind of hard.
Yeah.
Yeah. So it's not easy to break for some. So need to modify that also.
Yeah, we were trying once on a very soft paper, like the parchment or very soft. So the bite marks can be visible even on the wings, but then they are not stable in the moist weather, and they just
Yeah.
and then it doesn't work at all.
Yeah.
But the hard material didn't work well for us.
We found the scratch, and there were marks. This area trees, we don't have pictures here, but yeah, it is scratch. And at night is like raining, you know, this area humidity is like rain. That's how the trees get water, so it's very hot.
For those with UV reflect, how do you manage that stream?
Is that one? Yeah, it's actually surprisingly close. Well, I try different printers and I use Canon something laser printer with the original Canon ink. It print quite well. And also it's kind of water resistant for several days. You should go for laser printing. Otherwise it dissolves and disasters. Still it's experimental, yeah. I don't know. We thought of like using some other film, but then they cannot damage it. Anything else?
No, I think. it's
Yeah,
fine.
thanks,
Thank you
Liv.
very much. You will be here for more days, right?
Till I'm leaving to.
Oh, tomorrow, apparently tomorrow. Okay, so just a day. Thank you.